Hey gang, this is your video for section 3 of chapter 11, chapter 11 on cell communication, and the first picture we're looking at here is the big picture of basically how a signal gets from outside of a cell, gets received, and gets to the inside of a cell where it needs to be. And so what we talked about last video was the reception of a signal, and that reception basically, when that process, step number 1 here, is done, you're going to have gotten a signal that was received by a cell, and somehow that signal is now on the inside of a cell in one form or another. So typically that ended up with some sort of activated enzyme on the inside of a cell was the end of reception, and that was the signal, an activated enzyme on the inside of a cell. So now what we're going to pick up with is the transduction step, which is taking that signal that is just on the inside surface of the plasma membrane or has just gotten inside of the cell, taking that signal and somehow carrying it, maybe making it a larger and more common signal, but somehow carrying it to wherever the response needs to happen. Oftentimes it's the nucleus because we're turning on a gene, or maybe it's turning on some kind of proteins that will help a cell move, or turning on enzymes to make some kind of a protein. Anyway, so we're going to focus in on the transduction part of things. A term that is commonly used here is a phosphorylation cascade. And this is one type of transduction, is a phosphorylation cascade. It ends up being very, very common, but there are other types. We'll go through a couple other types as well, but just keep in mind that whatever the book is showing you here, it's honestly the tip of the iceberg as far as how transduction could happen, and this is just happens to be the most common. So anyway, this term cascade, I wanted to give you a visual on that. When they talk about a cascade, typically we're talking about a waterfall, if you were using it in common language. So here's a waterfall, and you probably wouldn't use cascade to describe like Niagara Falls, where it's just one huge drop, where you're at the very top of a waterfall and the water just boom straight down. It's going to be more like this one if we're using the term cascade, where there's the water's dropping from one level to the next, to the next, to the next, and it's hitting a rock and then bouncing to the next rock down and the next rock down and the next rock. That's a cascade, and this is kind of how if we draw out this type of transduction pathway, it's kind of like that, where in this case it's activated molecules are kind of bouncing to the next activated molecule, bouncing to the next one, and the next one, and the next one, and so on. So anyway, another thing before I even get into the diagram here, so that's that term cascade. The next one is something called a protein kinase. And a protein kinase, first of all, a kinase is an enzyme that phosphorylates something, typically taking phosphate from an ATP and putting it onto something else. I defined that in the last video, I believe. And then a protein kinase is just it adds a phosphate to a protein. Okay, so we're going to use a lot of those in this, and these are very, very common in the cell to have these protein kinases. Another term that shows up here is a relay molecule. Technically, all of these would be called relay molecules. They're carrying the signal from one place to another, or from one molecule to another. So where we typically would have left off after our first step, our reception, is we would have some form of a relay molecule made, whether it was an enzyme that was turned on or something else, but there'd be a relay molecule. And our transduction pathway is going to start when that relay molecule here basically joins an inactivated protein kinase. So it's just a protein kinase that is not functional. It's an enzyme, and it's not working. So when that relay molecule and this inactive protein kinase, when they join together, it activates the protein kinase so that it can do its job, and its job would be to put a phosphate on another protein. So then we have protein kinase 2 coming into the picture. Once we've activated protein kinase 1, if it can find a protein kinase 2, then it can activate it. The way it activates it is it joins it, that would be its substrate, and it will find an ATP, take a phosphate off the ATP, and put it onto protein kinase 2. Okay. Well, now we've activated protein kinase 2. It can do the same thing to another protein kinase. So then you'd have protein kinase 3 going through the exact same process. Protein kinase 3 going through the same process, and this could go on for a whole lot of steps. But eventually we're going to get to a protein kinase that its job is to phosphorylate some protein, usually an enzyme, but some protein that is required to start our cellular response, to actually do what we're supposed to do. Okay, then we have our active protein, and that'll go into our step 3, the cellular response. So this really isn't like a huge deal. It's kind of whispered down the lane type thing, where we're activating one, and it goes to the next one, activates that, goes to the next one, activates that. It's going to keep going until it gets to the goal, whatever that is, to turn on to some sort of a protein or activate some kind of a protein. The big question, and I'm actually going to talk about this at the end of I think video 4, section 4. The big question is why this seems so complex. Like why would we have this whisper down the lane type thing when we could just activate protein kinase 1 and then have that go and activate the initial or the final protein for the cellular response. And we'll get to that. There's a bunch of reasons for that. But yeah, just kind of quick overview on those is we can have more control over this process. The more steps there are, then the more places that we could control this. Other things are, we'll find that if we had one protein kinase 1, it has the ability to activate more than one protein kinase 2. So we can end up having large quantities until we get down through a bunch of steps. We've amplified, they call it, but we've made a whole lot of the protein kinase 3, which means we can activate a whole lot of the protein that needs to be activated for the cellular response. So that's one type, this phosphorylation cascade. It's a good picture of a transduction pathway. Now we'll go through some other concepts that come into these transduction pathways. Alright, the next thing to add to the picture here is something called a second messenger. A second messenger is going to be anything that's not a protein that's involved in a transduction pathway. So the one example that we're going to look at here, and then we'll look at another one of a second messenger, is AMP. So first of all, what AMP is, if we took ATP, the TP stands for triphosphate or 3 phosphates. If we take 2 of those phosphates off of this, then we make something called AMP, the M standing for mono. So monophosphate. So this second messenger is AMP, okay, and specifically it's called cyclic AMP, so we call it CAMP for cyclic AMP. But what happens here, first of all, we're showing in our response system, or our receptor system, we're showing a G protein linked receptor, and if you remember with the G protein linked receptor, it ends with an enzyme that has been activated. Okay, and here's a case where that enzyme that has been activated, its job is to turn ATP into AMP. Okay, so we have this activated enzyme here. It's called adenylyl cyclase, and if ATP hits this activated adenylyl cyclase, this enzyme, it'll produce cyclic AMP. Well, it ends up that cyclic AMP is the relay molecule that can turn on certain protein kinase cascades, phosphorylation cascades. And so there's just kind of adding a step to what we did on the previous slide, where we can make this cyclic AMP. It's not a protein, so we call it a second messenger, and it is the thing that activates our transduction pathway, so we can go down through this process of phosphorylating and dephosphorylating our protein kinases. So where are we at here? So that's cyclic AMP. Oh, the second messenger term, I don't appreciate it, because by definition a second messenger is anything in these transduction pathways that are not proteins. It does not mean that it's always the second step. In this diagram, we're showing that the first messenger, which this is true, the first messenger is the message that's coming from outside the cell. It's the first messenger. And then it's showing that our second messenger, and it kind of makes sense sequentially here that it would be our second messenger. Well, in the next slide, we're going to see that a second messenger could be third or fourth or fifth in the chain of transduction. It does not have to be the actual second step in the whole process. So anyway, but cyclic AMP, I think this is the most common second messenger, and it is used for all kinds of signals once we get the signal to the inside of the cell to get it to the protein kinases. So we'll look at the next one here. Okay, the next type of second messenger is an ion, a calcium ion. This also happens with sodium ions and some other things, but calcium ion. And for this particular pathway or this particular activity, I'm going to show you kind of an overview picture here first, just of what the typical situation is with calcium ions in a cell. And what happens is within a cell, typically calcium ions are pumped out of the cell pretty rapidly. So the cytoplasm or the cytosol of a cell is going to be low in calcium, and there's two places that these calcium ions get pumped. One place they pump it out of the cell, like Ed said. Another thing they do down here is they will pump calcium into different organelles, whether it's mitochondria over here or pump it into endoplasmic reticulum. But what we're doing is we're keeping a low concentration of calcium inside the cytosol. Okay, and that means there will be a higher concentration of calcium in the matrix of the mitochondria, or else in the inside of an endoplasmic reticulum. So that's the normal situation. What we're going to find is if a cell can start to allow the calcium in the cytosol to build up, when it does that, it can actually launch a transduction pathway. Okay, so that'll be kind of the steps there. So we'll look at the second picture here. This gets a little bit more detailed on this, or gets detailed on how the calcium is used as a second messenger. So what we're showing here is another G protein linked receptor, and this time it's going to be turning on an enzyme called a Phospholipase C enzyme. What Phospholipase C does, once it's activated at the end of our reception here, when it's activated, it will actually join something we're just going to call it PIP. I think somebody in class asked me, are we going to have to know what all these things mean, the PIP and the DAG and all that? No. I'm going to say don't memorize these things, but understand how the pathway works, if that makes sense. But anyway, it's going to take a PIP, it's a type of phospholipid, and it's going to break it in half. See these two molecules, DAG, don't worry about it, but the larger portion here is called IP3. Again, don't worry about what it stands for now, but that is a second messenger. It's going to carry the signal from the membrane here into the cell, and in this case, this second messenger is going to open up a gate protein. And so we're showing a gate protein here on the surface of the endoplasmic reticulum, and that gate protein, what it does is allows calcium to go from inside the endoplasmic reticulum back into the cytosol. So we open up this gate, the calcium then enters the cytosol, and it acts as another second messenger. So even though it's third in line here, it's going to act as another second messenger. And then those calcium ions, as they touch or attach to different proteins in the cell, they will activate them. So here's a way that the cell is increasing calcium concentration inside the cell. And when it does that, it starts to activate proteins that can cause a cellular response. All this time, we would be having calcium pumps pumping the calcium back across the membrane, which means relatively quickly we're going to get the cell back to its original state, and those proteins are going to be deactivated for the cellular response. And so if we want to continue to keep them active, we'd have to continue to make this IP3 so that it can open up the gate proteins so that calcium can come out and we can stay activated. So here's another second messenger. Again, this is an example where we're seeing second messenger, that term used, even though it's not the second step in this whole long deal. And I think that's it for this section in the book. So again, this is just taking the message from wherever the reception happened, whether it was on the cell membrane or within the cell, and it's carrying it to wherever the cellular response needs to be. So we saw a couple different examples here. And then our next video, we'll actually get to how the cell responds, as well as kind of reassessing the entire process and seeing where it is regulated and some things like that.